So, welcome back. Today, I'm going to talk to you briefly about cellular respiration. This is a process that goes hand in hand with photosynthesis, which we spoke about last time in the last video. So, what cellular respiration is the process by which living organisms break down sugar molecules and release that energy and use that energy in turn to make another molecule called ATP. So if you draw your attention to the board that we have in front of us, right, we see a chemical equation. C6H12O6, which if you recall is glucose, the sugar that was made by photosynthesis, plus O2 yields CO2 plus water plus ATP. Now, the reality of this equation should remove ATP. That should be off to the side because the ATP is not technically a product of this chemical reaction. Rather, it's something that is made using the energy released by this chemical reaction. Another little caveat is that not all cellular respiration requires oxygen. The cellular respiration that we do as eukaryotic organisms does, and cellular respiration that requires oxygen is called aerobic. Cellular respiration that doesn't require oxygen is called non-aerobic, right? Or anaerobic. Anaerobic. Yeah. So again, ultimately all this is organisms breaking down sugar to get the energy out. And I will talk in a moment about those steps that achieve that goal. So in order to understand what cellular respiration is doing, one must recall that this sugar molecule has within each of its bonds effectively stored the energy that was caught from the sunlight in photosynthesis. Right? Anytime you create bonds between elements to create molecules, there is a reserve of energy that is for practical purposes stored in those bonds. The reality of the storyline has to do with entropy changes and the availability of free energy versus used energy. And so to say that they're stored in the bonds is actually slightly inaccurate, but for pragmatic reasons, it's a good representation, right? So think of the energy as being stored in the bonds between these molecules. Cellular respiration rips apart these bonds and releases that energy and makes it available to do work. But it happens very methodically. So it happens step by step rather than happening in one big burst. And by doing it step by step, the organism can very carefully harness the energy that is released and use it to achieve very specific goals. And otherwise, all the energy would come out and smash out, and we wouldn't get much use out of it. Right? It's much more efficient to do it carefully and use the energy very specifically. So this sugar is broken down in three main steps through the process of cellular respiration. The first step is called glycolysis. Now, glycolysis is done by all living things. Super simple bacteria do glycolysis. It happens within the cytosol of the cell, and it requires no oxygen. And effectively, it's just taking the sugar and ripping it in half into two equal pieces. That process generates a little bit of ATP and then some byproducts called pyruvate. For more advanced organisms like us, we use those pyruvates to move into the next step. So the next step happens within the mitochondria, which, if you recall, is the organelle responsible for ATP production. Right? So this all ties together and makes all the sense in the world. So we're now in the mitochondria. We use those pyruvates to create two more high energy molecules called NADH and FADH2. Okay. These molecules are electron carrying molecules, and they're just temporary things that move the energy from one place to another within the mitochondria. Okay, so that's our second step, using the pyruvates to make these electron carrying molecules called the Krebs cycle. The third step and the final step of this whole process is the electron transport chain for oxidative phosphorylation. This is the step that makes the ATP. So it uses the energy that was carried in those electron carrying molecules, and it uses that energy to drive a mechanism that takes a phosphate and shoves it onto an ADP, phosphorylation. Okay. And that's how we get our ATPs. Right. And so, yeah, ta-da. In a moment, I'll move over to this drawing over here to explain where these steps are happening. So if we look at this drawing and we look at these numbers and these numbers, this is a diagram of a eukaryotic cell, and this is indicating where each of these steps is occurring. So glycolysis, again, is happening in the cytosol of the cell. It can happen anywhere in this area. Okay, this is just the gooey of the cell. The second step is happening within the mitochondria. So if you look at this organelle here, this is the mitochondria. An important anatomical structure of the mitochondria is the fact that it's double membraned. And so the Krebs cycle happens between the outside membrane and the inside membrane. Once it makes its products, it dumps those products into the inside of the mitochondria. And this is where that last and third step is going to happen. And this is where the ATP is ultimately made. As a general note about how this is working, there is a proton gradient created between these two membranes. And so a bunch of protons are outside and there's not much protons inside, and they want to move back in, but they can't. But there's a special gateway that lets them back in. And when they go through the special gateway, it spins the gateway. And that spinning of that gateway is called ATP synthase, is actually what's taking that phosphate group and physically shoving it onto the ADP to make ATP. So that is what the mitochondria is doing. That's what the Krebs cycle is doing. That's what the oxidative phosphorylation is doing, using the products from the glycolysis. And all three of those things together are what we consider to be cellular respiration. Okay. So just to wrap it up with an overview of the general principle about what's happening here, we're taking this sugar molecule that was made by photosynthesis, and we're using cellular respiration to break it down into smaller pieces and release the energy from the process of breaking it down. We are using that energy to take an ADP, which is this molecule right here, and we are shoving on a third phosphate group to make it ATP. Okay? Now the function of this molecule is that the cell will use it for work. So whenever a cell needs to do work stuff, such as pushing a molecule out of itself, it uses ATP. Right? This third bond holds a fair amount of energy, and so when the cell needs to do work, it rips off that third bond, and it uses that phosphate group to change the shape of whatever thing it is doing the work. So here I have a model that tries to depict what's happening. So for a very good instance of work is the active transport of molecules against a concentration gradient. So I have my protein that is going to be transporting this molecule out. Right? Before the ATP hits it, it does not have the right shape to move the molecule. You shove the ATP against it, that phosphate group leaves the ATP and joins on to this protein channel, changing its shape and doing the work of pushing out this molecule. Okay? And so that gives us the little bit of energy that we need to shove this molecule out and change the shape of this protein to make it work. After that's done, the phosphate group falls off, the protein reverts back to its original shape, and I am left with an ADP and a free phosphate that will eventually be going through this whole cycle of cellular respiration again to get rephosphorylated and to make more ATP. So yeah, that's it for cellular respiration. It is just how we get the energy out of sugar. So yeah, bye.